So the Medical School Lecture Committee, the Department of Neurosciences, and the Cleveland Brain Health Initiative are pleased to welcome today Dr. Thomas Sudoff to give the 2023 Louis A. Bloomfield Memorial Lecture, one of the two endowed lectureships here at the School of Medicine. And we welcome members of the Bloomfield family who are watching today online. Dr. Sudoff, as it states here, is an investigator of the Howard Hughes Medical Institute and is the Avram Goldstein Professor in the Department of Molecular and Cellular Physiology at Stanford University. In 2013, he won the Nobel Prize in Physiology or Medicine together with James Rothman and Randy Schekman, quote, for their discoveries of machinery regulating vesicle traffic. One aspect of winning the Nobel Prize is that you're asked to write an autobiographical essay, which is published on the Nobel Prize website. So rather than give a detailed description of Dr. Sudoff's many research accomplishments, I've decided to quote a few of his statements that I found raised important lessons about doing science. After mentioning that as a youth he learned to play the bassoon, he wrote, I credit my musical education with my dual appreciation for discipline and hard work on the one hand, for creativity on the other. I think trying to be marginally successful and learning how to be a musician taught me how to be a scientist. There is not creativity if one does not master the subject and pay exquisite attention to the details, but there is also no creativity if one cannot transcend the details. As to his research, he wrote, it is important to note that the nature of our studies was not revolutionary. In my career, no single major discovery changed the field all at once. Instead, our work progressed in incremental steps over 2 decades. I think this is a general property of scientific progress in understanding how something works. A single experiment rarely explains a major question, but usually a body of work is required. In terms of his studies with Reinhard Jahn on synaptic vesicles, he wrote, we set out to purify and clone every protein that might conceivably be involved and worry about their function later. This approach was initially criticized for being too descriptive, but turned out to be more fruitful than I could have hoped for, and has arguably led to a plausible understanding of neurotransmitter release. And finally, he wrote, quantity doesn't matter very much, nor does the place one publishes. In the end, what counts is discovery, which is often not immediately apparent. Most articles in so-called high impact, I put the so-called in, high impact journals, although highly cited initially, are soon forgotten, especially in our time, when it is basically the editor of a journal and not the reviewers who decides what gets published. An editor who often has limited knowledge of the subject but knows what is exciting. As scientists, we have to intellectually dissociate ourselves from fashion and journals and focus on what is the actual content of the study, what the data really say, not what the abstract says. So I think there's much for all of us to reflect here. But that's not why we're here. We're here to listen to Dr. Sudoff's lecture on the molecular machinery of synapse organization, neurexins. Dr. Sudoff.
Good afternoon, everybody. It's a pleasure for me to be here. I would like to thank Richard and everybody else who invited me for the invitation. And I would like to thank you for quoting those words that I wrote 10 years ago now. I still stand behind them, if ever more so. I think in science, and that's a long subject of discussion, we are in a difficult time period because it's no longer quite clear how we can reliably communicate what we actually believe, think, and discover. What I want to do today, however, is talk to you not about my rather peripheral thoughts on science, but actual data. And I want to talk to you specifically about the work that we've been doing in the last 10 years or so in trying to understand better how circuits are constructed. And as a way of introduction, you all know that the human brain is very complex. In fact, any brain is complex. There is a lot of neurons, trillions of neurons, and each neuron has thousands of synapses. When you compare that complexity to the complexity of the human genome looks quite simple. Nevertheless, we all know how difficult it has been to understand the human genome. So understanding the brain, how it is in the end conditioned, if not encoded by the genome, is a challenge. It's a challenge that neuroscientists have been grappling with at different levels, sometimes friendly, sometimes contentiously. But there's one concept that everybody shares at this point, which is this concept of the neural circuit. And what you see here is a microcircuit, could be anywhere, of two excitatory neurons in blue that are connected by synapses, and also to an inhibitory interneuron. This is a feed-forward inhibitory circuit. And what you can also see is an electron micrograph of a synapse. And it's the synapse that connects these neurons into circuits. It's the synapse that transfers information from one neuron to the next. And most importantly, it's a synapse that actually changes the information as it is being transported. So circuits really use synapses not just to go from one neuron to the next. Synapses are actually what changes the information in a way that depends completely on the properties of the synapses, which can be very, very different depending on what the pre- and post-synaptic partners are. What we have been trying to do over the last decade or so is to understand the molecular rules that determine the formation and specification of the properties of synapses. And the idea here is that synapses are junctions. They're junctions between two cells, similar possibly to other junctions between cells. And all intercellular junctions are organized by adhesion molecules. So we hypothesize that synapses are also organized by adhesion molecules. These adhesion molecules, however, don't primarily serve as mechanical devices. Instead, they signal. They basically signal. And they are asymmetric by nature because synapses are asymmetric. Synapses are very different pre- from post-synaptically, although there's many overlaps. In the end, as you can see just from this micrograph, there's a huge difference between the pre- and post-synaptic side. So the idea here is that there are synaptic adhesion molecules that are asymmetric in their interactions and that organize synapses. There have been many candidate synaptic adhesion molecules described, among which neurexins are arguably the most important, although others are probably as important. And that's what I'm going to talk about today, to try to tell you a bit about how we think neurexins organize synapses. I'm going to present a lot of data because I always do. Some of the data are in an abbreviated form. I'm not always going to talk to you about the controls, but I'm happy to explain them afterwards if needed, because I wanted to get through some concepts in my discussion with you here today. And so in my discussion, I will first introduce neurexins, because some of you may never have heard about it, which is inexcusable, of course. And then I will tell you about one particular project in detail for one particular facet of neurexins, of their function. In the last part of my talk, I will just give a glimpse at another function, just to give you a feeling for the whole thing before I end. So let me introduce neurexins. What you see here schematically is a synapse that illustrates neurexins on the left side and various ligands, a subset of postsynaptic adhesion molecules that they interact with on the right side. This is very schematic, it's very limited, but it gives you a feeling already just looking at this, that neurexins are multifaceted and that they interact with quite a few postsynaptic molecules, some of which I will discuss today. Neurexins are encoded by 3 genes, and each neurexin has 2 promoters. Each neurexin gene has 2 promoters, one for longer alpha neurexins that are shown here in this diagram like this, one for shorter beta neurexins. They are independently regulated in terms of their expression. Neurexins are localized at least in part, not completely, to synapses. This can be illustrated by immunocytochemistry here at the confocal level, showing a pretty close approximation to a presynaptic marker piccolo and a juxtaposition to a postsynaptic marker HOMA. We use DStorm in collaboration with Xiaowei Zhang at Harvard to localize where exactly in the synapse neurexins are. What you see here are samples of DStorm images where the red labeling corresponds to HOMA1, which is a postsynaptic marker, postsynaptic density marker, and the green signal corresponds to a neurexin that contains a knocked-in epitope tag because it's been difficult to get good antibodies. When you quantify these little dots, and what you can see here, what's interesting is that these dots always occupy a small bit of the synapse. They rarely cover the entire synapse. They are nanoclusters. And this is shown in better magnification here, either in culture or in tissue, culture, tissue. There's about 4 to 5 neurexins per nanocluster as measured by antibodies. And these nanoclusters are tiny. They're a couple of 100 nanometers as opposed to synapses that are somewhere between 500 to 1,000 nanometers. So neurexin clusters are forming a subdomain within synapses. What this looks like is that there's a single nanocluster, but a substantial part of synapses have 2 or sometimes even 3 nanoclusters. So there can be more than 1 nanocluster. Since we observed this some years ago, we have found the same thing for different presynaptic molecule tenuins, where you also have similar nanoclusters, and others in the field. Beautiful work from labs such as those of Tom Blanpeed have demonstrated that similar nanoclusters exist for other molecules as well. What is further interesting about neurexins is that there have been thousands of mutations, especially in neurexin 1, that's been observed in autism and schizophrenia, and actually Tourette's syndrome. These mutations are really interesting in that they are actually not point mutations, but they're deletions, and specifically they're CNVs, copy number variations. And they occur because the neurexin gene is humongous. It's several megabases long, and there's nothing else in this whole gene. There's sometimes introns are 500 kilobases, but there's no other gene. There's nothing. Okay? And so these different deletions, as you can see here in this overview, it's a subset of deletions that have been observed. They're not the same. They're all different. They're sometimes little pieces, sometimes big pieces, but they're always loss of function. They're heterozygous, and they're predisposed to a large number of neuropsychiatric disorders. So the question arises, how is it that a single gene can be associated with so many disorders and sometimes even be observed in apparently normal people? It raises the question whether or not this is meaningful at all, because very often you have genetic variation that can be more or less statistically significant, but maybe not that important. So some years ago, we tested that question using human neurons with two approaches, either an engineered conditional heterozygous neurexin 1 mutation or a patient-derived constitutive heterozygous neurexin 1 mutation. When you have neurexin 1 heterozygous mutations, the neurons develop just beautifully fine. We're using a method here that we developed about 10 years ago, induced neurons that I think many, many labs worldwide are now using with NGN2. It's widely used and very well characterized. It creates excitatory neurons. And what you can see here at the bottom is that the heterozygous deletion of neurexin 1 in this case causes a partial loss of expression, as you would expect. Functional effects. It turns out that there's a very discrete functional effect of this heterozygous mutation in these human neurons, and that there's a loss of synaptic strength, as shown here, corresponding to the decrease in mRNA levels, a loss that is due to a decrease in release probability. It likely, in fact, reflects impairment of a specific step, and so it intrigued us. It's intrigued us in many different ways because we wondered whether this had molecular correlates, whether there was a potential importance for patient-derived material, and why we hadn't seen something similar in mouse neurons. In terms of the molecular correlate, using extensive RNA-seq experiments, we found basically nothing. We found tons of changes that were highly significant statistically, but they were so small that biologically we chose to ignore them because we were doubtful, despite a statistical significance of 10 to the minus whatever, 30 or something like this, whether that has any meaning whatsoever. In terms of proteins, however, we found this one change that was not reflected in mRNA levels, which is an increase in the protein called Cas, a protein we discovered more than 20 years ago that binds to neurexins intracellularly. It's a presynaptic chemkinase MAGUC scaffolding protein. The function of this protein is as yet unknown. So this really, at that point, was an interesting question to us, which is this actually something that might be relevant for the disease? Is it possible that a general change in synaptic properties, a very discrete change in synaptic transmission, may be something that could, depending on genetic context, on genetic backgrounds in different people, predisposed to different diseases? And so we initiated a large-scale validation project to test the potential translational importance here. This is illustrated here. The first part of that project was, as shown here, to generate iPS cells from patients, then use our standard NGN2 protocol that we developed 10 years ago to make neurons, ion cells we call them, or induced neurons, a lot of people now call them, and then to do functional analysis in 2 different places to ensure that it's reproducible, that it's right. In addition to that, in parallel, we generated a new line of inducible neurexin 1 mutations, whereby CRE could cause the excision of exons that would cause a heterozygous deletion. And we compared that with the identical deletion in mice and tested whether these 2 systems provide similar data if you use exactly the same deletion and also exactly the same ion cell system, as opposed to using, let's say, primary cultures from the mice. With this approach, we found, first of all, that with the patient-derived neurons, we could fully replicate in different pairs of neurons versus of patients versus controls. Patients with a neurexin deletion, controls are matched as best as we could. We could fully replicate the phenotype. I'm not going to go through this in detail. The bottom line here is that this was replicated in independent places, demonstrating that there's a massive change in the initial release probability during a train of stimuli, but that it normalizes after multiple action potentials. So this is a very discrete phenotype that can be thought of as a selective synaptic impairment due to this heterozygous loss. We looked if this is also associated with a change in Cas protein, and it was, as shown here, very similar to the initially engineered cells that I just demonstrated to you a few minutes ago. We saw no other protein change as quantified here, except for an increase also in a protein called Chiad1, Chiad3, which is interesting because this protein is an enzyme that synthesizes quinurenic acid, glutamate receptor antagonist, which in turn, as you may know, has been at least by some evidence implicated in schizophrenia. So the overall phenotype of the heterozygous neorexin 1 mutation is much stronger than we had expected. In fact, when we started this project, we didn't think we would see anything in a heterozygous mutation because we hadn't seen something like this in mice. And so we wondered if this is human-specific. So as I already mentioned, we made exactly comparable neurons and analyzed them functionally. And when we did this, using exactly the same approaches, what we found is that we could again, this is now the third time, replicate the human neurons as MEPSC frequency with heterozygous mutations. This is the EPSC amplitude. You can see there's this massive decrease, and there's a paired pulse ratio demonstrating that this is a change in release probability. We have more evidence than that, just to illustrate this here. In the identically produced mouse neurons, we don't see that. It's just not there. There's no change in synaptic strength, no significant change. So in other words, neuron mouse and human neurons are somewhat different. Maybe you knew that already, but we had to convince ourselves of that. There is a huge difference in how they react, at least to some mutations. The bottom line here is that mice, as we all know, are great models for some fundamental processes, but they're not always reliable as disease models. So let me go on and tell you about the last feature, which will actually be a feature of neurexins that I will talk about a lot, which is their Baroque alternative splicing. This alternative splicing happens at 6 canonical sites in all 3 neurexins, except for one, where neurexin 2 is missing that site, and creates thousands of isoforms, as we and Peter Scheifele validated using biosequencing. And these thousands of isoforms are differentially expressed. Alternative splicing of neurexins is also important because it regulates many of their interactions, as shown here. For example, as I will come back to at the end of my talk, SS2, splice site number 2, only in alpha neurexins regulates binding to dystroglycan. Splice site number 4 regulates binding to an array of ligands, as shown here. So this is really what I want to talk to you about today in the main project, namely the question of what does alternative splicing that is highly regulated in neurexins actually do? And I'm focusing particularly on one splice variant, splice site number 4, because as I just showed you, that is the major determinant of many interactions of neurexins. Not all, but quite a few of them are regulated by this site of alternative splicing. And this just repeats what I just said. So the results, as I will tell you, and as is indicated in my summary slide here, is that these results of this study were actually quite unexpected. And I will try to explain to you why they were unexpected and what these results actually were. But to start at the beginning, in order to address this problem, we used a genetic approach because it's the most rigorous way of testing the importance of alternative splicing, especially of a molecule like neurexins, where there are many different splice variants, and where in rescue experiments you cannot really replicate all this alternative splicing. There's just too much. You can't have thousands of rescue constructs. Instead, we manipulated alternative splicing at splice head number 4 genetically by changing this exon 20, which is alternatively spliced. We changed the splice acceptor sequence from a non-canonical to a canonical one. As a result of this canonization, this exon is now constitutively included. So in the knock-in mice, this exon is always in. It's no longer spliced out. In addition to that, we flanked that exon with LOXP sites. So we could constitutively exclude it simply using Cre recombinase. In other words, the knock-in without Cre is constitutive inclusion, SS4 plus, with Cre is constitutive exclusion. SS4 minus, we use as a control a mutant Cre recombinase so that Cre and the control are always treated exactly the same. Okay. When we did this in cultured neurons initially 10 years ago, we were very surprised to observe for neurexin 3, which was this first neurexin gene we analyzed, that there was a decrease in AMPA receptor-mediated EPSCs in cultured neurons, but not in NMDA receptor EPSCs and not in iPSCs. We were surprised because this is the signature of a postsynaptic phenotype. Presynaptic release must be normal since NMDA receptor EPSCs are normal. But neurexins were thought to be presynaptic, not postsynaptic. Further work established that indeed there was a loss of postsynaptic AMPA receptor responses or postsynaptic AMPA receptors in the synapses in these cultured neurons, and raised the question whether neurexin 3 is really presynaptic, whether it acts presynaptically to regulate postsynaptic AMPA receptors. So we tested this in vivo. We used stereotactic manipulations in mice, in our mutant mice, at P21 to infect with AAVs, the CA1 region, as shown here. And then 2 weeks later, we cut slices and we did slice recordings. And we recorded from the subiculum, which is not infected, as you can see here. So we have only a presynaptic manipulation, and we stimulated the axons that emanate from CA1 neurons to innervate the subiculum neurons. And this preparation allows the analysis of specifically presynaptic effects on synaptic strength and plasticity. And when we did this, we confirmed that there was this selective decrease in AMPA receptor-mediated responses, as shown here. The control, when you have presynaptic SS4 minus with Cre, is normal compared to an unrelated wild type, whereas with a presynaptic SS4 plus, there is a decrease. There was no effect on MDA receptor-mediated responses. So this demonstrated that presynaptic neurexin 3 alternative splicing regulates postsynaptic AMPA receptors in the subiculum. Neurexins are very similar to each other. The 1, 2, 3 genes, similar, alternatively spliced, very homologous sequences. We assumed that neurexins 1 and 2 would have the same functions, that they would all be redundant functionally. And so it took us a while to come around to analyze neurexins 1 and 2. In fact, it took me a while to analyze, to convince a postdoc to actually analyze another neurexin, because nobody wants to do this more of the same, right? But I finally convinced Jinya Dai to do these experiments, and she analyzed neurexin 1 SS4 plus minus mice in the same paradigm. There was no effect on AMPA receptors, as you can see here, but now there was an effect on MDA receptors. Totally different phenotype. Moreover, the effect was opposite. Now SS4 plus increases the response, doesn't suppress it, increases, which is normalized in the presynaptic excision of SS4 in neurexin 1. We also analyzed neurexin 2. There was no effect whatsoever, either SS4 minus or SS4 plus. No effect on AMPA and MDA receptor responses in this particular synapse. So unexpectedly, the 3 neurexins are intrinsically different. And we performed a number of additional experiments that I would like not to spend the time on right now to show that this is the case, including overexpression of the various neurexins and so on. There's an intrinsic difference between the three neurexins. This is quite amazing. And that leads us to the conclusion that presynaptic SS4 alternative splicing of neurexin 1 regulates NMDA receptors such that SS4 plus dominantly upregulates it, whereas presynaptic alternative splicing of neurexin 3 regulates AMPA receptors and downregulates them. This is a mechanism of transsynaptic receptor control whereby presynaptic neurexin alternative splicing instructs the postsynaptic neuron. How can we think about this? How might this work? And this is what I'm going to talk to you about in the next part of my discussion. I'm going to tell you basically how much we know at this point how this works. And the starting point here is that since it's the SS4 plus form that makes the action, the SS4 minus form is inert, it must be a ligand that specifically only binds to SS4 plus. And it turns out that there's only one ligand known at present that only binds to SS4 plus neurexins, and that are cerebellins, which are C1q molecules, as shown here, that come into two classes, cerebellin 4 versus 1, 2, 3, because they bind to different receptors. So we wondered whether these might be a responsible expression mapping identified. Cerebellin 2 is the only cerebellin that's expressed in the subiculum. So we generated cerebellin 2 knockout mice and did the same experiments that I just showed you. We first checked for changes in synapse numbers, because if you read the literature, you will find that it is an established fact that cerebellins do synapse formation. So we wondered whether the knockout would change synapse numbers. And as you can see here, there's no change in synapse numbers. The cerebellin knockout has no effect on synapse numbers or on spine density. Actually, in nowhere except in the cerebellum could we find any change on spine numbers or spine density with any cerebellum. So, then we measured synaptic transmission, slice physiology, AMPA receptors and NMDA receptors. When we knock out cerebellin 2, AMPA receptor responses go up. SS4 plus makes it go down for neurexin 3, and NMDA receptor responses go down. SS4 plus of neurexin 1 makes it go up. So it's the mirror image of the neurexin manipulations of both of them together. It's exactly what you would expect if cerebellin 2 was the effector. You're basically removing the effector of the SS4 plus variants of neurexin 1 and neurexin 3, and thereby have the opposite phenotype that they have. Okay? Do these changes represent the same synapses, or are they separate synapses? To test this, we used minimal stimulation, again in slices. Everything up to now is in slices. And you can see that in the minimal stimulation experiments, using the same paradigm, we see changes in the same, presumably the same synapses, suggesting that these changes actually happen in the same synapses and not in separate synapses. So cerebellin 2 regulates both AMPA and NMDA receptors at individual synapses. How does this work? Now it turns out that cerebellin 2 has as a receptor that's well characterized the so-called GLU-D molecules that are also known as GRID1 and GRID2 by their gene symbols, or as GLU-R delta 1 and GLU-R delta 2. And they're called this GLU whatever because they resemble glutamate receptors. They are homologous to AMPA and NMDA receptors that are homologous to each other. But they are not glutamate receptors. They don't act as glutamate receptors. So this suggested the binding, this receptor binding here, suggested to others. We didn't do this work, unfortunately. It's really beautiful work primarily from the Mishina and Yuzaki labs in Japan, that there is an interaction, a transsynaptic interaction. And thus, the GLU-Ds are the prime candidates for an effector of the cerebellin 2 neurexin complexes. So we deleted GLU-D1, but since GLU-D2 is not highly expressed in the subiculum, this work is now done in culture using CRISPR. Everybody does it. Again, we measured synapse densities, no change. Maybe not a surprise at this point. No change in the density of synapses when you knock out GLUD1. We then did the physiology. It phenocopies the cerebellum 2 knockout phenotype. It's the same phenotype as shown here. AMPA receptors go up, NMDA receptors go down, indicating that these are truly, as you would almost expect, the effectors. GLUD1 is the effector for cerebellum 2 and for the neurexins. And we also did in vivo experiments confirming these results. Moreover, we did double mutant experiments of GLUD with cerebellums and neurexins in various combinations, which confirmed this pathway. So what I've tried to tell you at this point is that neurexin 1 and neurexin 3, differentially regulate NMDA and AMPA receptors in opposite directions as SS4+, via, surprisingly, the same transduction module, the same module that they bind to, presynaptically, transsynaptically. This is a hard thing to imagine. It really makes you wonder, how is it possible that presynaptic neurexins like this with SS4+ binding to the same module can have different postsynaptic responses? And this is something we grappled with. And we hypothesized that for this to work, the GLUDs must have different effector pathways for NMDA and AMPA receptors that are differentially stimulated, because otherwise it wouldn't work. There must be something in GLUD molecularly that either regulates NMDA receptors or AMPA receptors. And so what we did is we identified conserved sequences in GLUD1 and GLUD2, cytoplasmic tails. We mutated these sequences and we tested whether such mutations would impair rescue of the GLUD knockout. And what you see here are responses of AMPA receptor and NMDA receptors, EPSCs, in cultured neurons again with a GLUD1 knockout, demonstrating what we observed in these rescue experiments. Let me first focus on the AMPA receptor EPSCs. As you can see, we got the same phenotype, thankfully. The GLUD1 knockout, yeah, it's always worrisome, you always wonder. Yeah. The GLUD1 knockout causes this increase in AMPA receptor responses because you block the break induced by the presynaptic neurexin 3 SS4+. This is rescued by all constructs of GLUD1 except for one, mutant number 4, which is a mutation in motif number 4. When we look at the NMDA receptor response, again, reproduces the suppression of the response in the knockout. Again, all the constructs rescue except mutant number 3. So in other words, different mutations in the cytoplasmic tail impair the rescues, confirming that there must be separate pathways via GLUD1 that regulate either AMPA or NMDA receptor responses. And these changes, by the way, correspond to changes in the surface levels of AMPA receptors and NMDA receptors. So what we end up with here is illustrated schematically here, whereby SS4 plus of neurexin 1 and SS4 plus of neurexin 3 converge on the same transduction module. GLUD1 and cerebellum 2 in the case of the subiculum. And this then translates into differential responses, either an upregulation of NMDA receptors or suppression of AMPA receptors, whereby the presynaptic neurexin 1 or 3 differential signal must be sensed differentially by the GLUD molecules. One fascinating feature, at least for me personally, from an evolutionary perspective, is that GLUDs are actually homologous, evolutionarily related to AMPA and NMDA receptors. And we know very well how AMPA and NMDA receptors are regulated, how they actually work. And they work via a change in the conformation of the transmembrane regions, which opens up a channel. That is the mechanism of signal transduction. So we wondered whether GluD1 binding of cell balance might also cause a change in the transmembrane architecture, thereby leading to a change in some way of the intracellular cytoplasmic tail. Please.
Yeah, I have a Synapse 101 question. I presume, not being in the field, that the same synapse can express different neurexins. If so, then what would be the specificity, the presynaptic specificity, that dictates whether you have one or the other type of neurexin being expressed?
It's a great question. So the same neuron expresses, nearly all neurons express all neurexins, but in very different amounts. Alternative splicing at SS4 for each neurexin is separately regulated by a mechanism I don't know. So each neuron actually has a characteristic pattern of neurexin expression, both in terms of the levels of the principal isoforms and of their alternative splicing.
And the other question, from a biochemical viewpoint, I'm interested. The LEL sequence is highly negatively charged. What's the significance of the sequence?
I'm sorry, which sequence?
Does LEL mean leucine, glutamic acid leucine? Is that what it means?
Which LEL?
You have these sequences LELLLELL. No, on your cartoon. Yeah. The N terminus, the C terminus. Oh yeah, the N terminus. Yeah.
Oh, I'm sorry, I didn't go into that detail. Okay, I'm sorry. Yeah, so maybe I should have explained the structure of neurexins in detail. I'm sorry. So neurexins are large molecules. They're multi-domain proteins. They're typical adhesion molecules. Most adhesion molecules have multiple domains. In the case of neurexins, the L stands for LNS domain, which is a very widely distributed in nature. The E stands for EGF-like repeat. So this is not a sequence. This is a domain. So this whole thing is about 150 kilodaltons, and it's sort of a string of domains. Okay. I'm sorry. Yeah. I have to learn. I should have explained this in more greater detail. They're typical adhesion molecules, and the beta neurexins just have one LNS domain. That's the only thing they have, the 6th LNS domain, which is the one that's alternatively spliced. Okay. My apologies. Yeah. Okay. So, could GluD1 operate by a transmembrane conformational change like AMP receptors and MD receptors? So to test this, we used a very reductionist approach. I like reductionist approach. I try to, in my work, always to reduce everything to the simplest possible level. And so what we did is we simply took out all of the transmembrane regions and substituted them for the single transmembrane region of CD4, which is, as you know, an immune cell receptor. And we took also out the ligand binding domain of GluD1 and kept only the N-terminal domain because that's what binds cerebellum. So you have a molecule that has only the N-terminal cerebellum binding motif and the cytoplasmic tail. GluD1, like all glutamate receptors, is a tetramer. This is a dimer because this domain forms a dimer by itself. We then tested whether this would rescue the phenotypes of the GluD1 deletions. And we did the same thing also for GluD2 because we wanted to know whether they were redundant functionally. What you can see here in this experiment, in this rescue experiment, is very simple. All of them rescue. Both GluD1 and GluD2 with a single transmembrane region rescues just perfectly fine, either the increase in AMPA receptors or the decrease in NMDA receptors. So the bottom line here is you actually don't need that whole transmembrane stuff that's left over from evolution. All you need, presumably, is cerebellum binding outside that translates into an intracellular signal that regulates AMPA and NMDA receptors. Now, the final question here that immediately comes up, obviously, is whether in this particular construct here, it is again the same sequences in the cytoplasmic tail that are required for this type of regulation. And we first addressed that with point mutations in the cytoplasmic tail, which suggested that this is the case, and which data I'm not going to show you. And then we went all the way out and did an even more reductionist approach. We simply deleted the entire cytoplasmic tail in this very reduced molecule and inserted in an otherwise unrelated sequence these motifs that were from these mutated sequences as shown here. These minimal constructs contain only N-terminally cerebellum binding domain of GLUD1 connected to the C-terminal cytoplasmic tail, somewhere between 4 to 8 residues. And when we tested these in rescue experiments, as you can see here, for the AMPA receptor immunoresponses, again, we have the same typical increase in AMPA receptor immunoresponses with a GLUD1 knockout. There's only one construct that rescues, which has the 4A motif. If you put a point mutation into the 4A motif, it no longer rescues, confirming that this motif needs to be in this sequence in order to rescue. Conversely, with the NMDA receptor, you have the suppression of the NMDA receptor responses. There's only one construct that rescues, motif 3. All the others don't rescue. So very short cytoplasmic sequence motifs fully regulate AMPA receptors and NMDA receptors in a manner controlled by neurexin cerebellum signaling. In other words, what emerges here is a relatively simple way of regulating AMPA and NMDA receptor responses in a synapse, whereby a presynaptic input causes a postsynaptic differential response dependent on whether it's neurexin 1 or neurexin 3. This response is mediated by cerebellums binding to GLUD that then causes an effector signal. Now, there's a number of questions you might raise at this point. How is this possible that this presynaptic signal is differentially translated? And how is it possible that without a conformational change, you get this kind of response, kind of regulation? We hypothesize that this is done by multimerization because cerebellums are not monomers, they're multimers, dimers. And as such, they would cross-link GLUD molecules into arrays. We think that this explains a potential, provides a potential avenue for explanation, which unfortunately is difficult to test because without the multimerization sequence, they don't bind to neurexins. So you can't just monomerize them. We also hypothesize that the differential effects are mediated by differential affinities for cerebellum. This is based on the fact that neurexins have differential affinities for cerebellums, different neurexins, which might explain why either NMDA or AMPA receptors are regulated. However, that's where we stand at this project, which suggests that at least one pathway that shapes the receptor complement of a synapse involves a presynaptic regulation of postsynaptic receptor contents via neurexins. I just want to emphasize that that's not the only way. NMDA and AMPA receptor abundance in a synapse is maybe one of the most important features of synapses. Synapses differ dramatically in how much receptors they have, and it is thus important to keep this under big control. As many of you probably know, for example, LTP changes that ratio. And it's interesting that, maybe not surprisingly, the neurexin 3 knockout blocks LTP. presumably because it interferes with that pathway. Let me, in my final 10 minutes or so, just give you a glimpse of yet another function of neurexins to make sure you don't go home with the impression that neurexins are there to regulate AMPA and NMDA receptors. They are at those synapses where there's cell balance in GLUD1 or GLUD2. But they do many other things. In recent work, for example, we discovered that cereblin 4 in a complex with neogenin 1 in a completely different synapse, perforant path synapse in the dentate gyrus, is essential for LTP, but not for basic synaptic transmission or for NMDA AMPA receptors. But this is not what I wanted to talk about in my last minutes here. Instead, I wanted to tell you about a completely different function that is regulated by splice site number 2. And that is the regulation of the presynaptic release probability by neurexins. The first phenotype, the first function of neurexins that we observed many years ago was to regulate presynaptic release probability. And that is still the one function that is most widely found in synapses. What I'm going to tell you about today, however, is a one particular project in one particular synapse that illustrates a mechanism, which again, in my view, is somewhat surprising. And this was studied for neurexin 3, first in olfactory bulb synapses, and specifically in dendrodendritic synapses in the olfactory bulb, and later on in other synapses as well. The initial experiment was performed in cultured olfactory bulb neurons that form dendrodendritic synapses even in culture. The advantage of the system is that you can identify the mitral cells very easily. They're huge. You can patch them. You can identify granule cells. You can patch them too. And so you can look at synapses between these cells in a reduced system. And when we did this for straight neurexin 3 knockouts, these are not the SS4 manipulated neurons. These are straight neurexin 3 knockouts. What we found was that there was no change in AMPA or NMDA receptor EPSCs. And in fact, the SS4 mutants had no effect on either one in this particular synapse because there is no cereblin 2 at this point. But that the iPSC was decreased. And further work demonstrated that this decrease in iPSC amplitude was due to a decrease in release probability. So we were interested in trying to understand how this works as an example, as a sort of one case at least, where neurexins are essential for a normal release probability. And the effect was robust, which makes it easier to work on an effect. And over the years, we performed rescue experiments that try to understand what is required for this effect, what is required to have a normal release probability, what part of neurexin 3 is required. I'm not going to tell you the entire story because I want to focus on the bottom line, which was achieved by doing rescue experiments with either full-length neurexin 3 or with neurexin 3 alpha LNS2 domain only. And so I apologize. Now the numbers here correspond to different LNS domains, which are called L in my diagram. Okay. This A, B, C is the EGF domains, the first, second, and third. These are the 6 LNS domains. And the LSS2 is in the second LNS domain. And it has 3 splice variants, minus 2A and 2AB. And so what I'm going to tell you are rescue experiments only with this construct, because that turned out to be decisive in understanding. And what you see here are rescue experiments in slices now from in vivo manipulations, granule cell to mitral cell synapses in dendrodendritic synapses, recorded with extracellular stimulation. And what you can see is if you have delta Cre, that's the wild type, that's the input-output curve, Phi causes about 50% decrease in responses. This can be fully rescued just with this tiny construct here, just one LNS domain, SS2 minus, but not SS2 plus. So this was a surprise to us, and this is actually a release probability. This is just a coefficient of variation, but you could use also paired pulse ratios and other measures of release probability. So this surprised us because it suggested that release probability, which is presynaptic property, depends on some extracellular feature of the neurexin, suggesting that this is not cell autonomous. That in other words, the neurexin that is expressed in the cell doesn't cell autonomously regulate the release, but has to interact with something else in order to do this. In other words, again, these results reveal that SS2 alternative splicing controls the presynaptic release probability, but it was puzzling because I had always thought that presynaptic would be a release would be a cell autonomous property of neurexins. And so this is SS2, and the only known ligand that binds exclusively to SS2 minus is dystroglycan, which is a cell adhesion molecule that's particularly well characterized in muscle and blood vessels. It has many functions in the body, but is also present in inhibitory synapses. So it has many functions. There's other molecules that bind to SS2 plus and minus, including a family of proteins called neurexophilins. But they bind to both, and so they couldn't explain it. We identified this complex of neurexins with dystroglycan many, many years ago, where we showed that only SS2 minus bounded and that this was calcium dependent. And so we tested with CRISPR deletions if the dystroglycan deletion in vivo would do the same thing. And as you can see here, this is the case. If you have dystroglycan deletion, CRISPRing it out, you get the same decrease, and this is now postsynaptic in the mitral cells. Coefficient of variation goes up because the release probability goes down. So it phenocopies the presynaptic deletion of neurexin 3 or the SS2 function of neurexin 3. So what this then suggests, in other words, is that neurexins at this particular synapse and a few other synapses, certainly, maybe more, regulates the presynaptic release probability not by a cell autonomous mechanism, but by binding to dystroglycan, which then in turn, by an unknown mechanism, I have no idea how this works, feeds back onto the presynaptic side to mediate the postsynaptic regulation of neurotransmitter release. And the same, as I already mentioned, we found that the same mechanism operates in a subset of inhibitory synapses in the cerebral cortex. So this is just another one of the functions of neurexin signaling. I wanted to tell you about this, as I said, to make sure you don't walk away from here with the idea that neurexins, that is a simple case of doing one thing. No. I like to visualize neurexins as signaling platforms that resemble a Swiss Army knife. They're platforms with different features. And you might think of these different features of a Swiss Army knife like the LNS and EGF-like domains of the neurexins that are in my diagram, my cartoon. And that each one of these has different roles. So if this is a neurexin, one may be involved in recruiting calcium channels to the active zone. One must regulate postsynaptic AMPA receptors and NMDA receptors, presynaptic receptors, presynaptic PK channels, haven't told you the data, LTP, haven't told you the data, GABA receptors, endocannabinoid synthesis. All of these have been connected to neurexins. This is important because, as I tried to explain at the very beginning, synapses differ dramatically in properties. And properties are many. It's not just release probability. It's not strength, whatever strength means. It's short-term plasticity, long-term plasticity, ratio of AMPA to NMDA receptors, and, with different compositions. So, as such, then, neurexins are central to conditioning how the synapse operates in a circuit. And this is also important, I believe, for neuropsychiatric disorders, because it's interesting, as you probably know, that at least in schizophrenia, NMDA receptor pathways have been very much implicated in the pathogenesis. There are mutations on NMDA receptors extremely rare that predispose to schizophrenia. So there's a pathway here that connects neurexins to other molecules that have been implicated in the pathogenesis. The way this works, we believe, is that various neurexins isoforms specify synapse properties via competing ligand interactions. Often the same domains bind to different ligands, depending on splice variants and affinities and abundance. I refer to this as the dynamic interaction network. And in this manner, neurexins may contribute to the molecular logic that constructs synapses and neural circuits. I'm not going to go into this. So let me close. It's exactly 5 o'clock. Let me close by acknowledging the people who did this work. So the work that I just discussed on the GLUD1 cerebellum neurexin axis that regulates AMPA and NMDA receptors was largely carried out by Jinya Dai, who just left and started her own lab at Mount Sinai in New York City. The work on the dystroglycan neurexin 3 connection was performed by Justin Trotter and Cosmos Wang, who is now doing his postdoc at Harvard. And Men Zhang and Lulu Chen and Jason Aoto made essential contributions to earlier stages of the project. I am grateful to Xiaowei Zhang for the collaboration on the D-Storm, which was really eye-opening to me. And I am even more grateful for the funding that we need, because otherwise we couldn't do any science. Thank you very much. Happy to take any questions.
Thank you very much for a clear lecture. Could you explain how the electrical activity of the synapse in relationship to LTP changes the expression of neurotoxins?
Yeah, so that is a great question, because the dynamics of gene expression and alternative splicing are now becoming more and more a center of attention, especially as a function of activity. And at present, there is data in the literature that looks very compelling, but has not yet been reproduced independently, suggesting that alternative splicing of neurexins may be activity dependent. So there may be a change in alternative splicing in neurexins. What is on more firmer ground, because it has been independently observed in experiments that are much easier to perform, because it's much easier to study gene expression than alternative splicing, is that cerebellum expression, at least cerebellum 2 and 4, is highly dependent on activity. So it goes up with activity. It's activity dependent. Thus, there's at least the possibility that expression of components of the system do serve to restructure synapses in an activity-dependent manner. A separate question is how this system intersects with classical LTP. We've worked a lot on the mechanism of LTP. And LTP is no longer of general interest, but it still fascinates me as a phenomenon that is unexplained and whose significance, to the best of my knowledge, is unclear. We still don't know what LTP does, nor do we know really how it works. Yeah, it changes AMPA receptors, as first demonstrated by Roberto Marano. But what does it actually mean? And how does it work? And, so surprisingly, multiple ligands of neurexins, as well as neurexins themselves, are required for LTP in the sense that they don't just decrease LTP. I'm always suspicious when there's a decrease or increase in LTP. They block LTP completely. There's no LTP. How that works, I have no idea. But it just illustrates how little we actually understand how LTP really is generated. And there's very few people who work on this these days anymore. So I'm not sure we'll ever find out. Yeah, at least not soon. Yeah. Go ahead. Yes.
So earlier, if mice weren't a reliable model to study the relationship between neurexins and the disease, was it okay to study all the alternative splicing related things to it? Did you run into any problems?
Yeah, great question. So maybe all we are studying are rodent-specific mechanisms that may never happen in a human. Yeah, you know, that's a risk we have to take. In general, in biology, there are rules that are kept and rules that change. And we won't be able to tell one from the other until we know the rules. So it is absolutely possible that in the human context, these mechanisms are different or shifted or the magnitude is different. Yeah, it is possible. And we will find out, hopefully, at some point. Yeah, it's a good question. But we have to figure out the rules first. We'll never be able to otherwise tell. Yeah. And we work a lot with human neurons these days because it's a very straightforward system. It's to study fundamental mechanisms in a very reductionist level.
The days I was working on acetylcholine receptors in my first postdoc, so thank you for the update. So the question is, you showed that several hundreds of mutations/deletions are really associated with various pathologies. Now, what about the reverse, gain of function, creating a super synapse? Have you ever observed gain of functions?
No. So this particular locus, there's no duplications known. It's too big. Well, it could be potentially if you had the whole thing, yeah. But this is a very large gene, especially neurexin 1 and neurexin 3 are huge genes. They have lots of repetitive elements, so that's why you have all these deletions. But if you had duplications in these stretches, you would also get a loss of function, because for obvious reasons, right? Because it's not the whole gene. So different from, for example, 16p CNVs, where you have both deletions and duplications that lead to slightly different clinical associations. In this case, that isn't the case. There's only deletions known. Yeah. It's an interesting question. Yeah. And the next time I give this talk, I'll remember that when I introduce neurexins, that I should explain what, yeah, I'm sorry, yeah. You know, that's the problem of the thing. When you work on something intensely, you tend to forget what other people don't know.
So you remember us, Kelly here has produced a plaque for you to take back.
Thank you so much.